As I put my lecture together, I reflected on the state of our field, and a recurring but disturbing theme emerged. We screwed up. We live in a world awash with antibiotic-resistant bacteria. Diseases once subdued have now returned with a vengeance. The previously vibrant field of antibiotic research and development is now dying. Unless something is done soon, very soon, devastation is just around the corner. The decimation of the antibiotic development infrastructure and our inability to use antibiotics to enable the miracles of modern medicine. But despite all this, I believe we have it within our power to begin the world again. Doctors Ehrlich, Domak, and Fleming, you entrusted us with real-life magic bullets, but we didn't fully understand them. In your time, the three leading causes of death, pneumonia, tuberculosis, and diarrhea, were all bacterial diseases. But after your discoveries, those diseases didn't even register in the top 10. In your time, 80% of bacterial pneumonia patients died in America. But thanks to your work, that number dropped from 80% to 5%. It felt like we were given superpowers. We thought it was time to close the book on infectious diseases. Nobel laureate Dr. Sir Frank McFarlane Burnett summed it up best when he said, "One can think of the middle of the 20th century as the end to one of the most important social revolutions in history. The virtual elimination of infectious diseases as a significant factor in social life." If he could only see us now. In our zeal, we prescribed antibiotics to patients who did not need them. Up to 1/2 of the annual 150 million antibiotic prescriptions are not necessary. We used antibiotics to treat diseases that didn't call for it, like acute viral bronchitis. And when the disease did call for antibiotics, we underdosed them. And even when we used them correctly, we used them for too long. We even polluted the environment with them. So to Doctors Ehrlich, Domak, and Fleming, while we were grateful for the superpowers you gave us, we're also sorry how we mishandled them. We screwed up. But I believe all is not lost. I believe we have it within our power to begin the world again. That's a quote from Thomas Paine, the American revolutionary. Fitting because I see a revolution on the horizon. Our view of antibiotic use and misuse has been changing. Antibiotic stewardship is the focus of clinicians, hospital administrators, medical societies, government, and the World Health Organization. The goal of these programs is to ensure that patients receive the right antibiotic, the right dosing regimen, and the right treatment duration. From this, you might think that we finally turned the corner. But in reality, things are not so rosy. In a survey of 192 U.S. hospitals in a recent CDC study published in JAMA, found that antibiotic treatment was unsupported in nearly 56% of patients. 56% of antibiotic use was unsupported. Believe it or not, that's the good news. What's the bad news? Antibiotic misuse is likely much, much worse, largely because antibiotic stewardship faces so many challenges. First, the programs are overly focused on restricting antibiotics due to an effort to save new antibiotic drugs for the treatment of resistant bacteria. Secondly, clinicians lack tools to guide the selection of an optimal drug, dose, and duration of treatment. And lastly, antibiotic stewardship is based on outdated models. As an example, let's look at clafran, also known as cefotaxime. Cefotaxime was discovered and developed in the 1970s and became available for clinical use in 1981. At that time, cefotaxime was one of the most widely investigated antibiotics in the world. It was the first of a new, third generation of cephalosporins. and it was stable against the most prevalent beta-lactamases produced by Enterobacteriaceae at that time. Everyone was very excited about it. We thought that it was the cephalosporin that would literally make a world of difference. And shortly thereafter, other third-generation cephalosporins came to market, such as ceftriaxone, also known as Rocephin. Everything was great for a short time, because by the late 1980s, we started seeing some worrisome reports of increased clinical failure and mortality associated with third-generation cephalosporins. It turns out these patients were infected with Enterobacteriaceae producing extended-spectrum beta-lactamase, also known as ESBLs. Worse yet, a significant percentage of ESBL-producing strains were not being detected by hospital clinical microbiology laboratories. It was a dire situation. For instance, Melzer and colleagues reported a statistically higher mortality in those patients infected with ESBL-producing Enterobacteriaceae compared to those who were not. Even more concerning is that there was no consensus as to why the situation had occurred and what to do about it. Most people thought testing was the main problem. So there was a strong emphasis on the development of ESBL screening tests that hospital clinical microbiology laboratories could use to detect these strains. However, there was a small group, Doctors William Craig, Ronald Jones, Mike Dudley, Sujata Bhavnani, and I, that believed the true cause as to why some ESBL-infected patients did not respond to third-generation therapy was based in pharmacology. We also believed that we could detect ESBL-producing strains without increasing the burden on understaffed clinical microbiology laboratories. We searched the literature and conducted laboratory studies for many months until a critical insight came from Dr. Craig. And what was that clinical insight? Have a look at this figure that he and Dr. Andes generated in the neutropenic murine thigh infection model. It was not the presence or absence of infection with an ESBL-producing isolate that predicted response. Rather, it was the percentage of the dosing interval that drug concentrations remained above the MIC. And while it was true that ESBL-producing strains have higher MIC values, the time required that drug concentrations remained above the MIC did not change with ESBL production. This insight was a paradigm shift. That is, efficacy was predicted by drug exposure indexed to MIC and not ESBL production, as many people thought. So instead of developing cumbersome and costly ESBL screening tests, perhaps the only solution we needed was to simply correct the third-generation cephalosporin susceptibility breakpoints. PK/PD targeted attainment analyses were then conducted by Drs. Bhavnani, Dudley, and me in the context of surveillance data provided by Dr. Ron Jones. These analyses identified new breakpoints designed to discriminate between pathogens likely to respond to third-generation therapy from those that would not. The new breakpoints were much lower than those supported by NCCLS and the FDA at that time, and there was pushback. Many screamed, "Who are we to change these breakpoints?" saying the breakpoints were supported by clinical data from randomized clinical trials. They had a good point, but we felt there was something missing. So Drs. Craig, Bhavnani, Dudley, Jones, and I set out to re-examine the clinical data that supported the breakpoints for older drugs like cefotaxime. And we discovered a number of problems. These are data from cefotaxime-treated patients that were included in the original new drug application submitted to the FDA and published by Clyde Thornsberry and colleagues in Reviews of Infectious Diseases in 1982. The data set is pooled from a wide variety of clinical indications, including bloodstream infections, soft tissue infections, urinary tract, intra-abdominal, and gynecological infections, where the doses ranged wildly from 0.5 gram to 12 grams per day. It was noted that responses were generally high for Enterobacteriaceae and gram-positive cocci and lower for Pseudomonas aeruginosa. These clinical data were viewed in the context of epidemiologic data. These cefotaxime MIC distribution data are based on a collection of nearly 8,300 isolates and published by Fuchs and colleagues in AAC in 1980. Note that the isolate collection forms three distinct distributions. Enterobacteriaceae are shifted to the left, Pseudomonas and Acinetobacter are shifted to the right, and methicillin-susceptible Staph aureus are in the middle. In an effort to have the same susceptibility breakpoint for two major pathogen groups, the data for Enterobacteriaceae and Staph aureus were pooled to support a susceptibility breakpoint of 8 milligrams per liter. These are the pharmacokinetic data used to support the cefotaxime Enterobacteriaceae breakpoint decision, as published by Clyde Thornsby and colleagues in Reviews of Infectious Diseases in 1982. They stated, and I quote, "These data show that the usual dosage of cefotaxime can produce blood levels that are adequate for the inhibition of most isolates of Enterobacteriaceae. But remember, most Enterobacteriaceae isolates have MIC values of 0.5 or less. Had they added a horizontal line using the breakpoint value of 8 milligrams per ml, they likely would have noted that very few patients had exposure sufficient for efficacy." We had our answer. The breakpoints were indeed the problem. The original NCCLS and FDA Enterobacteriaceae susceptibility breakpoints were not supported by robust clinical and PK/PD data, only pooled data across different bacterial species and wildly different clinical indications and wildly different dosing regimens. An approach we would never consider valid today. In 2004, after almost a quarter century of clinically, pharmacologically, and epidemiologically unsupported cefotaxime Enterobacteriaceae susceptibility breakpoints, CLSI began debating the need for breakpoint changes. It took another 8 years of debate at CLSI to make a decision to correct the cefotaxime Enterobacteriaceae susceptibility breakpoints to less than or equal to 1 for susceptible, 2 for intermediate, and greater than or equal 4 for resistant. In all, it took over 30 years to recognize and correct the problem. In that 30-year interval, exacerbated by poorly conceived breakpoints and selection pressure, third-generation cephalosporins came to dominate therapy for Enterobacteriaceae infections, and ESBLs came to dominate third-generation cephalosporins. It makes you wonder how different things would be if we had gotten it right from the start. The Clafran story is likely not unique. The majority of older intravenous IV antibiotics have not had their breakpoints reevaluated using today's standards. But what if they were? I believe not only would this enable us to make better use of our older antibiotics, but also allow new antibiotics to thrive. It's no secret that our field today has witnessed a near complete market failure of new antibiotics, despite the fact that new antibiotics are sorely needed. The reimbursement model for antibiotics plays a part, but I believe they're not the whole story. Let's take a look at plazomicin, also known as Zemdry, which was developed in the 2010s. It was marketed as the aminoglycoside that helps lead the way out, presumably out of a forest of multi-drug resistant Enterobacteriaceae. Achaogen spent around $1 billion to develop plazomicin, a total that includes nearly $125 million of government funding from the Biomedical Advanced Research and Development Authority, also known as BARDA. Achaogen finally launched plazomicin into the market in July of 2018. But only 9 months later, Achaogen filed for bankruptcy and sold the rights for $15 million. Let that sink in. An investment of over $1 billion resulted in a value of only $15 million, less than a year after approval. So the big question is why? Plazomicin was developed specifically to meet the unmet medical need for drugs to treat ESBL-producing Enterobacteriaceae and carbapenem-resistant Enterobacteriaceae, like KPC. Plazomicin even received qualified infectious disease product status by the FDA. So what happened? The first thing you need to understand is that the PK/PD of plazomicin is similar to that of older aminoglycosides like amikacin, gentamicin, and tobramycin. Here are neutropenic murine thigh infection model generated by Dr. Bill Craig. Note that amikacin, gentamicin, and tobramycin all display similar PK/PD profiles. Here, plazomicin data generated by Dr. George Trusano and Arnold Louis are overlaid on that for the old aminoglycosides. Despite these sets being generated 2 to 3 decades apart in different laboratories, you can clearly see that plazomicin doesn't look all that different from the older aminoglycosides. It's the same story for KPC-producing isolates. Here's the same data except with KPC producers highlighted. But you may be asking, what about ESBLs and carbapenemase-producing isolates? The unmet medical need that plazomicin was specifically designed for. Even here, activity appears similar between plazomicin and amikacin against ESBL producers. For this reason, clinicians in the market could be forgiven for overlooking plazomicin in favor of the similar, established, cheaper aminoglycosides like amikacin. So that's what happened. Case closed. Or is it? I believe there's more to this story. I believe that things could have been very different for plazomicin and amikacin had amikacin's breakpoints been looked at more closely. The breakpoint differences between amikacin and plazomicin are striking. Amikacin's breakpoints were established in the mid-1970s against a very different set of standards than we use today. But what if we evaluated amikacin in the exact same way plazomicin was evaluated by the CLSI and FDA? It turns out that amikacin looks very different. Here we see the probability of PK/PD target attainment for amikacin overlaid by its Enterobacteriaceae MIC distribution. First note, at the current breakpoint at 16 milligrams per liter, target attainment is 0. Let that sink in. At the amikacin susceptibility breakpoint of 16 milligrams per liter, the probability of attaining an effective drug exposure is 0. What breakpoint is actually supported? These data support an amikacin Enterobacteriaceae susceptible breakpoint of 1 to 2 milligrams per liter. Contrast that with the plot on the right for plazomicin. These data support a plazomicin Enterobacteriaceae susceptible breakpoint of 1 milligram per liter. Assuming an amikacin 20 milligram per kilogram every 24-hour dosing regimen, 81% of all Enterobacteriaceae isolates would be categorized as amikacin susceptible with a corrected breakpoint of less than or equal to 2 milligrams per liter, rather than 99% with the current FDA and CLSI breakpoint of 16 milligrams per liter. But this is where it gets even more interesting. Remember this table that showed similar activity between plazomicin and amikacin against ESBL producers? This is what it looks like after applying appropriate amikacin breakpoints. Using the contemporary dose of 20 milligrams per kilogram daily, amikacin susceptibility drops to 53% and 24% in ESBL and carbapenem-producing isolates, respectively. Remember that plazomicin was developed specifically to meet the unmet medical need for drugs to treat ESBL and carbapenemase-producing isolates. And that's exactly what was delivered. But it was amikacin's outdated and incorrect breakpoints that oversold amikacin's abilities and masked the true value of plazomicin. How would plazomicin and amikacin have fared had we reevaluated older drugs under the same PK/PD standards? I believe, if we had, we'd be providing better patient care, have more investment in a more vibrant antibiotic development industry than we have today. So far, we've seen how many problems have stemmed from outdated models we use to test older drugs. But imagine how much worse it would be if we didn't test at all. Well, believe it or not, even now, we use and prescribe antibiotics that are not routinely tested in clinical laboratories. Let's look at cephalexin, also known as Keflex. Cephalexin was discovered and developed in the 1960s and became available for clinical use around 1970. It was the first oral cephalosporin antibiotic and was stable against the most prevalent penicillinases produced by Staphylococcus aureus and Enterobacteriaceae at that time. And it provided an alternative to first-generation penicillins and aminopenicillins, which were no longer reliably active against these pathogens. Everyone was really excited about Keflex. Even now, Keflex is the third most prescribed antibiotic in the United States, but we don't routinely test its activity in clinical laboratories. Our perceptions of its activity and many other oral antibiotics is through the lens of surrogate testing. That is, if an isolate is susceptible to antibiotic A, then the isolate is considered susceptible to antibiotics B, C, D, and E. In the 1960s, while Eli Lilly was developing cephalexin, they also developed two additional cephalosporins, cefazolin and cephaloridine, an agent administered IM. Folks at that time felt it would be much simpler to test one agent rather than three. So they chose cefazolin, or cephalothin, which was another early cephalosporin developed in the 1960s, as a surrogate for cephalexin. It was a nice idea, but not one well-grounded in reality. I mean, think about it. There is a four-fold difference in potency against E. coli, which favors cefazolin over cephalexin. There's also a large difference in pharmacokinetics. For instance, the peak serum concentration following a typical 1 gram cefazolin dose is 185 milligrams per liter, which is 10 times that typically observed following a 500 milligram cephalexin dose. And cefazolin has a longer serum half-life. The notion that an isolate categorized as cefazolin susceptible will magically respond to therapy with a less potent agent that has a weaker pharmacokinetic profile is wishful thinking. But as mentioned earlier, this notion was from a different era. These decisions were made literally BC. No, not this guy. This guy. PK/PD principles and concepts were not understood in the 1960s. Those would not begin to come into focus until the 1980s, the life's work of Dr. Craig. Given the lack of historical PK/PD data in the literature, we decided to study cephalexin in a modern in vivo infection model. The model we selected was a neutropenic pyelonephritis model, and the challenge isolates were each considered susceptible to cephalexin according to the surrogate testing with cefazolin. If these isolates were truly susceptible to cephalexin, then we should see substantial bacterial killing in infected animals. Here we see the cephalexin data from the murine pyelonephritis model. First note that cephalexin efficacy is not well described by percent time above MIC. I say that because even at 100% time above MIC, response to therapy is unpredictable. Second note that cephalexin efficacy is much better described by another time-dependent PK/PD measure. That is AUC to MIC ratio times 1/tau. Finally note that as we drive cephalexin exposure higher, there are diminishing bactericidal effects. Even at the highest cephalexin exposure study, the end result was no change in bacterial density at the end of therapy relative to the start of therapy. Remember, these E. coli isolates are considered by CLSI as being susceptible. How do these cephalexin data compare to new antibiotics? Here are the tebipenem data from the same murine pyelonephritis model. Notice how as tebipenem exposure increases, so too does its bactericidal activity over a wide range of exposures. This is what a reliably effective antibiotic looks like. It should be evident from these data that the CLSI surrogate test is not an accurate appraisal of cephalexin's in vivo activity. Even if you try looking for evidence of the drug's clinical efficacy in historical urinary tract infection studies, you won't be impressed. Here's one example. These cephalexin data are from a study that was conducted by Eli Lilly and the National Institutes of Allergy and Infectious Diseases and published by Marvin Truck and colleagues in Antimicrobial Agents and Chemotherapy in 1969. By the end of cephalexin treatment, the urine was rendered sterile in only 67% of patients with E. coli in their urine. What kind of clinical and microbiological efficacy at the end of therapy do we expect for new antibiotics? 90% or greater. When one looks across the totality of clinical trial data involving cephalexin and urinary tract infections, you'll note three things. Many studies are not well controlled, they are small, and have highly variable results. One has to wonder if decades of oral beta-lactam selection pressure driven by inappropriate surrogate testing has facilitated the evolution of a wide spectrum of beta-lactamases that now threaten every class of beta-lactams. Maybe the root cause of our global resistance problem lies not in the IV bottle, but rather in the tens of millions of pills and capsules administered to patients worldwide, which is facilitated by poorly conceived surrogate breakpoint testing. But remember, susceptibility to an antibiotic alone is not the optimal predictor of treatment outcome. The basis for this statement is that when MIC alone is considered, we leave out a lot of other information that is critical to predict treatment success. To predict treatment success, one must integrate a variety of types of information that spans the microbiology in animal laboratories through clinical data. Two of the most important factors to integrate are drug exposure and MIC. Let's look at tigecycline-treated patients with complicated intra-abdominal infections. Here we see bacteriologic response by pathogen MIC. Note that we do not see a clear pattern of more treatment failures as MIC increases. Now let's look at response by AUC. Again, we don't see response increase as AUC gets greater. Finally, let's look at response by AUC to MIC ratio. The pattern of failure becomes evident. Failure occurs most frequently among patients with lower exposures, as described by the AUC to MIC ratio. We see these types of relationships in vitro and animal infection models for every antibiotic we've studied. We see these types of relationships in individual clinical trial data sets and across multiple NDA programs. That is, as PK/PD target attainment goes up, so too does the probability of good things. In this case, the probability of a new antibiotic meeting its primary efficacy endpoints and being approved by drug regulators. I believe that worldwide, the way we currently support antibiotic treatment decisions is broken. Patients all too often receive the wrong antibiotic or the right one dosed poorly. Sure, there's therapeutic drug monitoring for a few antibiotics, but you can count them on one hand and make spare change. It's really hard for physicians to adopt new tools. It requires a dramatic change in how they practice medicine. And they don't want to stop and type a bunch of stuff into a computer. They really want to focus on the patient, and that's a good thing. We need better tools. To illustrate, let's go to grand rounds together at a virtual hospital near you. GLD is a 72-year-old, well-nourished male with type 2 diabetes and COPD who presents to the hospital with community-acquired pneumonia. He is intubated in the emergency department, admitted to the ICU, and started on an empiric regimen of ceftriaxone and levofloxacin. On day 7 of hospitalization, things take a turn for the worse. His oxygenation requirements worsen. New pulmonary infiltrates are identified on his chest x-ray, and a tracheal aspirate was obtained, and a gram stain showed many white blood cells with a non-lactose fermenting bacteria. A preliminary diagnosis of late-onset ventilator-associated bacterial pneumonia was made, and the patient was started on piperacillin tazobactam, 4.5 grams, every 6 hours, infused over 4 hours, based on an estimated creatinine clearance of 86 mLs per minute. 2 days later, the patient's culture returned positive for Pseudomonas aeruginosa, which was susceptible to piperacillin tazobactam and also cefepime and ciprofloxacin and meropenem, but resistant to tobramycin. Piperacillin tazobactam was continued. The patient's condition initially improves, but then worsens. 5 days later, on day 14 of hospitalization, the patient is recultured, and his antibiotic was changed to cefepime, 2 grams intravenously, infused over 30 minutes every 12 hours. The patient's condition deteriorates rapidly. The patient dies 4 days later. The final respiratory culture result, obtained shortly before death, grew multi-drug resistant Pseudomonas. Sadly, the case presented is not a corner case, rather it is the norm. 1.5 million patients enter the hospital with a primary diagnosis of pneumonia every year. Some will progress to ventilator-associated pneumonia, of which Pseudomonas is the leading cause. Pseudomonas is one of the top healthcare-associated pathogens and is considered a serious threat by the Centers for Disease Control and Prevention. This pathogen has a remarkable capacity to develop resistance to commonly used antibiotics, such as carbapenems, fluoroquinolones, aminoglycosides, through various resistance mechanisms that may be expressed simultaneously. Antibiotic resistant strains are relatively common. 1 in 5 Pseudomonas infections in the United States involve a multi-drug resistant isolate. More than 32,000 cases and 2,700 deaths are attributed to MDR Pseudomonas each year. So how could things have been different for our patient? What was the mistake? Was the pathogen susceptible to the initial antibiotic, piperacillin tazobactam? Yes. Was the pathogen susceptible to the second antibiotic, cefepime? Yes. Did the patient get dosed according to standard and reasonable treatment guidelines? Yes. Was it the physician's fault? No. Was it the stewardship pharmacist's fault? No. What went wrong then? The mistake was not taking into account variability. What do I mean? Anytime you give a dose of an antibiotic to a patient, there is a probability distribution describing a wide range of possible exposures. Similarly, for every antibiotic pathogen pair, there is a distribution of pathogen susceptibility to that drug. At the end of the day, there is no one-size-fits-all effective dosing regimen. And there is no such thing as a susceptibility breakpoint that reliably forecasts efficacy for every patient. It is only when we integrate these probability distributions that we overcome variability and identify the most likely effective regimens. Imagine if we had a PK/PD optimized antibiotic decision support platform that integrates into the clinician's workflow with a background feedback process. Let's go back to our patient, specifically to the day the decision was made to initiate piperacillin tazobactam therapy for Pseudomonas aeruginosa. The tool could integrate the diagnosis. This tool could use population pharmacokinetic models and leverage information readily available in the electronic medical record like patient characteristics, age, gender, height, weight, chemistry information like serum creatinine and creatinine clearance, microbiology data like pathogen identification, susceptibility test results, antibiograms, to conduct a series of complex calculations using simple and complex differential equations that integrate patient pathogen-specific factors and their associated variability to simulate tens of thousands of possible outcomes. The result? A level of clinical decision support that was never attainable before, all done behind the scenes in the blink of an eye without slowing the clinical workflow. Here we go. Notice that while the performance of the standard piperacillin and tazobactam regimen was improved with a 4-hour infusion, the probability of attaining an effective exposure in our patient was not so hot. And when the patient's condition deteriorated, his therapy was switched to cefepime. And that regimen's performance was even worse. In fact, the microbiology laboratory data indicated that the isolate was susceptible to piperacillin, tazobactam, cefepime, and ciprofloxacin. But in reality, given this patient's specific characteristics, the likelihood of attaining an effective exposure with standard doses of these antibiotics in this patient was poor. The clinician was blind to the differences between these therapeutic options. Because of the paradigm in which we make our antibiotic treatment decisions is broken. As you can see, there were better options available to the physician. One has to wonder what would have happened if the patient received PK/PD optimized therapy right from the beginning. I believe chances are his condition may well have improved. He would have spent less time in the resource-intensive ICU, and he would have returned to a normal daily life. This EMR integrated precision dosing decision support platform is actually something we've been working on for a number of years. I hope to be able to share it with you and the community very soon. While I feel like we screwed up, I also believe it's not too late. In fact, we have an opportunity to regain our superpowers. If we fix the problems with breakpoints, surrogate testing, and outdated tools, I see a new world on the horizon. A world with a wide selection of antibiotics to choose from, both old and new. A world with a vibrant antibiotic development infrastructure. A world where bacterial resistance is slowed and antibiotics continue to enable the miracles of modern medicine. I believe we have it within our power to begin the world again. The American Society for Microbiology is the oldest single life science organization in the world. Many distinguished scientists and no less than 10 Nobel Prize winners have addressed this august gathering. Being selected to give the ASM antibiotic lecture, also known as the ICEC lecture, is an honor with few parallels. I would like to close by saying thank you. Thank you to Dr. David Andes and the selection committee and the American Microbiology Society. And thank you to some very special and incredible people. Doctors Richard Quintiliani and Charles Nightingale. The best educational decision I ever made in my lifetime was entering your postdoctoral fellowship program. Charlie, you taught me about life, integrity, and how to be a leader. Dick, you challenged me to live in a creative place. You both provided me with a framework, a toolbox, that included the powerful lens of PK/PD, with which to view the world, to ask and answer questions, make discoveries, and early on in my career, annoy every single person around me with the PK/PD superpowers you bestowed upon me. Dick and Charlie, thank you. Doctors Bill Craig, George Trusano, and Ron Jones, you took me under your collective wings and helped my career grow exponentially. Your contributions to the field of infectious disease chemotherapy are simply amazing. Bill, for your lifetime of work, you deserve the Nobel. George, you are the single most insightful person I have ever known. Our understanding of the conditions needed to suppress antibacterial and antiviral resistance are owed to you. Ron, you are an icon. Your superb scientific and leadership qualities are worthy of emulation. Thank you. Doctors Christopher Rubino and Sujata Bhavnani. Together, we founded the Institute for Clinical Pharmacodynamics. Our goal was simple yet audacious: to change the way antibiotics are developed and used to care for patients worldwide. I say without hesitation that if it were not for you two, I would not be giving this lecture today. In large measure, this lecture belongs to each of you. You earned it as much or more than I. Thank you. I love you both. And to my wife, Sherry, my cancer survivor, you gave me this space, the freedom to take risks, and the time to work harder than most thought wise. You gave me unending support, and most valuable of all, a wonderful family to come home to. And finally, thank you all for listening today.